Hello and good morning, wherever you may be. My name is Sarah Jane Demy of Demy-Colton, and on behalf of the Demy-Colton team, our sponsor, and all of our supporters, it is my pleasure to welcome you to the 23rd installment of the Demy-Colton Virtual Salon Series. Today, we will focus on humans versus machines. We'll get to that in a moment. We welcome all of our past participants back again, and of course, we welcome everybody who is new to the virtual salon series. Demy-Colton's mission is all about convening healthcare industry leaders from multiple disciplines to catalyze change. And we typically do this through in-person events in the healthcare space. And of course, as with you, COVID changed everything about what we do and forced us to reconsider how we deliver. That led to the birth of the Demy-Colton Virtual Salon Series, which will be here to stay when we return to the new normal. I encourage everybody to check our website for any scheduled upcoming salons. And if you have ideas for salons that you would like to see us do, always feel free to reach out to us. On another note, I am pleased to let you all know that we will be launching a new conference here in New York City, BioFuture, on October 5 and 6. It will take place at Cure, Deerfield Partners' new virtual innovation space over on Park Avenue South. And needless to say, we are just really excited about our ability to launch BioFuture at Cure. It was delayed last year because of COVID. Before we get underway, I'd just like to thank our sponsors today, Turea Partners, for today's salon, and all of our past BioFuture sponsors and our founding sponsors. Some housekeeping notes before I turn the virtual podium over to our moderator and panelists. The structure of today's program will be a 50 to 55 minute formal discussion, which will be followed by a Q&A session. You are invited to submit your questions in the Ask a Question pane below the screen, and we will get to as many as possible. Also, please note that this salon is being recorded and will be available on demand later today. Additional materials can be found in the Event Resources tab. I'm freezing, so please know that it's really hot here in New York City, as I'm sure it is elsewhere, and that may be leading to some of the technical issues that we may be having. Bear with us, please. Now to today's salon, humans versus machines. Before I introduce the moderator and the panelists, this salon is the first of its kind competition to predict clinical trial success between artificial intelligence generated predictions as well as human generated predictions. We'll get more, our moderator will get to that a little bit further on. So today is the first installment of a 4-part series. The second installment will take place at BioFuture, where there will be the readout of the success of these predictions, and as well as an introduction of the next group of assets that will be reviewed and read out during Biotech Showcase later in the year, or at the beginning of next year, I should say. Our moderator today is Dr. Lee Nguyen-Jatko. Lee is Executive Director of Pharma Custom Intelligence at Informa, and our panelists are Daniel Chancellor, Director of Thought Leadership, Informa Pharma Intelligence. We have Mark Gordon. He's the founder and CEO of Diviner, the human component of this competition. Dr. Matthias Koff, Head of Product Management at Molecular Health GmbH. Dr. Daniel Meitelka, Head of Methodology at Diviner. And Friedrich von Bohlen, CEO and co-founder of Molecular Health GmbH. So without further ado, let me turn the virtual podium over to Lee. Lee, take it away.
Thank you so much, Sarah. And thank you everyone for your time today. I'm really excited to be moderating this panel where we get to deep dive into different ways to predict clinical trials outcome. So as you may know, it takes about 10 and a half years from drugs in phase 1 to make it to regulatory approval. And data within our Informer's Biomed Tracker database shows that less than 8% of drugs that are in phase 1 actually make it to the market, and that the biggest hurdle is actually the phase 2 component of the studies, where less than 30% of candidates actually pass phase 2 studies and phase 2 status. So clearly, the clinical trials outcome in phase 2 determines that go, no-go decisions for a lot of drugs. And predicting how these clinical trials will do is of the utmost importance, so that not only can companies determine which assets to go forth with, but also take attention away from those that may not be successful to concentrate on those that can be successful, so that we can bring medicines to patients sooner. So that's my take on why this is so important. But I want to pass it over to our panelists so that they can also talk about why predicting clinical trials outcome is so important. And if I can start first with Dan Chancellor from Informer, just to give that sort of more global and independent view as to why this is important, that would be great. Over to you, Dan.
Super, thanks, Lee. I'm really excited to be part of this panel. I'm going to try and be as impartial as possible, that independent moderator between the AI and the human, although obviously trying not to side too long with the humans. My background is predominantly on the commercial strategy and decision making. At Informer, we also have tools and products that help clinical and regulatory decision making. Some of the numbers that Lee has just mentioned come from a paper that we've recently published in collaboration with Bio. 9 out of 10 drugs entering phase 1 will ultimately fail. Phase 2 is the biggest hurdle. But within this, there are success stories. There are ways in which you can manipulate or game the system or design drugs more intelligently, design clinical trials more intelligently. And we want to be spreading the words as far and wide as possible to kind of espouse the best practices that drug developers can employ to develop not only drugs with the highest success rates, so in a sense, gaming the system to generate positive trials is one aspect of it, but also running clinical trials in such a way that's going to produce really meaningful clinical results and ultimately benefit patients at the end. So I'm interested in a way from the predicting whether a clinical trial is going to work or not, but ultimately it's designing the best trials to produce the best drugs and the best quality of evidence. And anything we can learn from our colleagues on the panel, whether it's from an AI algorithm kind of recommendations or human intelligence recommendations, I think we should be sponging up as much as we can, learning as much from the contrasting approaches where the similarities are, and ultimately try and design the best drugs possible.
Thank you, Dan. And perhaps I could pass it over to Friedrich for your take on why this is so important.
Hi, Lee. Thanks everyone for joining this interesting contest. Let me give you a little background here on molecular health, that you understand where we come from, and then answering your question. Basically, we're a precision medicine company with a mission to deliver unique computing technologies for precise molecular understanding, to decode diseases, for individualized diagnosis, treatment, improved patient outcome, and drug development. For now more than a decade, we have systematically developed what we call DataOme, in analogy to genome or proteome. So DataOme is our global clinical knowledge data platform. It empowers integration and transformation of clinical and molecular data into real-life applications for better decision-making in therapy, drug discovery, drug development, drug commercialization, and drug safety. On top of DataOme, we have developed applications for therapy decision support and better drug development. One of these applications is MHPredict, that we will learn more about here today. And we started to develop MHPredict about 3 years ago for a contest of an investment bank. The question of that bank was, can someone, and can we, molecular, precisely predict and accurately predict the outcome of clinical trials? Simply speaking, to give you a little background, MHPredict merges clinicaltrials.gov with our DataOme and uses gradient boosting-based models for the prediction of clinical trial success. Our models are trained on more than 40,000 trials with known outcome, each with around 200 features from drugs, diseases, pathways, trial design, sponsors, and many more diverse other features that we found important and supportive for our predictions. In that contest with that bank, we correctly predicted 84% of trials given to us by the bank, since our overall accuracy is at around 80%. So why is that so important? It is, of course, so important for the reasons David mentioned before. I mean, still one of the biggest challenges in drug development is the high dropout rates of projects, with all the costs and other, let's say, frustrations around it. And we think that improving the success rates of clinical trials will lead to many different advantages. I mean, drugs can be developed faster, they can develop with better success rates. That means lower amortizations, which means at the end lower drug costs. For patients on trials, it means that they have a higher likelihood that the trial they're at and they're on is successful, so really helps these patients, so to say, in development already. So I mean, long story short, better trial prediction will lead to better drugs faster, safer to the market, and for investors, it will allow to pick successful companies and projects better, which is also important and an improvement for investors and their businesses.
Thank you so much, Friedrich. That was great. And Mark, if I can pass it over to you to take Diviner's take on this.
Sure. Thanks, Lee. Thanks, Sarah, for putting this event together, and Lee and Dan for moderating. You've all raised some really interesting points. One other way of looking at the problem is if you consider assessing the value of a drug in development, the factor that gets the least attention because it has the most uncertainty is the probability of success, more so than revenue and costs. And if the industry is going to bring more transformative therapies to market and to patients, we're going to have to get a lot better at assessing probability of success. There are two main ways, as we see it, to tackle this problem. One is the big data, machine learning algorithm approach that Friedrich and Molecular Health are working on. They're doing fantastic work there. And another approach, a second approach, both we think are important, are to tackle cognitive human biases and flaws in traditional forecasting processes. So the last decade has shown tremendous advances in this area with human-based techniques in widely cited published papers by groups such as the Good Judgment Project. So what are these techniques? There's a few aspects to them. One big surprise is that deep domain expertise does not make somebody a good forecaster. Forecasting is a skill, and some are better at it than others. Another is that if you step back and consider the outside view as the exact opposite of what you want when you're in a data room, it does lead to better predictive results. And a third is if you apply structured processes to identify, train, and leverage the right blend of niche experts, domain experts, and broad skilled forecasters, you get better predictive results. So we founded Diviner last year to create this blend of techniques and apply it to drug development, to predicting drug development outcomes and their impact on stock prices. We're developing a proprietary method, which Dan will talk a little bit more about, Dan Nytelka, in a minute. And we're really excited about how far we've come. We have a broad team of diverse drug development and assessment experts, forecasting science, and in the past few months, while we're raising our seed funding, we've recruited a diverse team of 25 industry experts who are intrigued about this challenge and about sharing in our upside. We've predicted 13 trial outcomes, 10 of which are in this event today, and we're expanding our network and our methods and really excited about sharing some of these ideas.
Thank you so much, Mark. So let's get into it then. So the purpose of this competition, as Sarah has mentioned earlier, is really looking at AI-driven forecasting and human-led forecasting to predict clinical trials outcome. And we've done this we have 10 that the Molecular Health and Diviner team has done. It is posted in a document which is attached to this presentation. If you can look at it, you can see the 10 predictions. It should be noted these predictions were done before any trial outcome has read out, and there is one trial that has read out, so we'll take a look at that later. But before we get into sort of the deep dives of it, I wanted to get Dan C's take, the independent view of just that overall trend of what he sees as the main differences and similarities between the AI approach versus the human approach. So Dan C.
Sure, thanks, Lee. So if you can bring up the event resources tab and have the handouts. Ideally, this would be on a piece of paper and we'd be all live, but unfortunately, we have to make do with the digital routes. Some really kind of big observations that we've made from of the contest as a whole. We've not been involved with any of the predictions. We're purely judging based on what we're seeing in front of us, and humans are much more optimistic than the algorithm. 8 out of the 10 clinical trials that are part of this contest, the humans have assigned a probability of success higher than 50%, so we can class that as this trial will succeed. The 2 trials where the human approach, the Diviner approach, were less than 50%, they're still pretty close to that, 50-50, just I think 40% or so. And if you and these are both phase 2 trials, if you factor in that phase 2 success rates are only about 30%, even for these negative trials, there's still quite a generous, well, I won't say generous, but a positive assessment of, likelihood of showing a positive outcome. They're also more tightly bunched. But if we consider the algorithm on the flip side is more negative. 8 out of the 10 predictions suggest that this clinical trial will not succeed. The definition of that being it won't meet its primary endpoints. And among some of these predictions, they very much tend towards the extremities, including quite a few very, very low percentage predictions. And I'm really interested to understand what some of the features and characteristics that the algorithm is considering that contribute towards such a low success rate. So with these kind of basic observations, I want to turn this into some questions for our panelists. And I want to start first with molecular health. Why is it you think that the algorithm is more critical and also tending more to the extremes? So I think at this stage, Matthias, do you want to come in for this question?
The algorithm is generally more negative, as you stated. I think it's typical to humans that they don't really tend to do predictions in the extremes. So that's one of the factors. And a lot of the negative predictions are from small companies. And we looked at the features, of course, that were the key drivers for potential failure, according to our algorithm. And many of those companies seem to be rather unexperienced, so they don't have many phase 3 trials running or had run them in the past. And the drugs that are investigated, a rather low number of trials were ran for those drugs. So it seems to be a combination of rather experienced-looking companies from the perspective of the AI and pretty new stuff. And that's definitely 2 very significant factors that push the predictions towards failure. But those are not the only factors. I mean, there's depending on the trials, there are other factors that already also have a negative influence on that. Yeah. And I mean, generally, I'm sure some of those companies, they have highly experienced people and they may not be so unexperienced as they may look from the AI's perspective. That's something that humans with a lot of work will probably dig out, and they might have some advantages on that side. So from that perspective, it's always good to look at the AI predictions, at the most impactful features, and then to judge yourself if you want to base a critical decision on the AI outcome, or if you say, okay, I don't trust this prediction, and all that transparency is provided in our software. So it's not just a single value that you get, but you get feature exploration, you get reliability charts that allow you to assess the reliability of the prediction. And so it's really an AI tool that's best used by human experts in the end.
Okay. Maybe before I start questioning you on some of that, we'll give Dan M a chance to discuss what some of the factors were that sort of made the human predictions somewhat more positive.
Well, let me comment first on the phase 2 probability of success. And as both Yuli and Daniel mentioned, the typical quoted phase 2 probability of success is more in the 30%, 35% range than in the range of a lot of the trials that we're looking at. But let me distinguish them a little bit in that what we're doing is we're predicting individual trials, which is different than the phase transition probabilities. So if you look at an overall phase 2 program, it's going to have at least a couple of trials and maybe more. You might have a 2A, then you can have a 2B, and so on. And so what you really have to do is look at the combined success of all of the trials in the phase 2 to determine whether you're transitioning to phase 3. So we might have a higher than typical probability comparing against the phase probability, but for an individual trial, you'd expect a significantly higher probability of success. As Matthias pointed out, we do look at a lot more detail that's very specific to an individual opportunity. So we're looking at any information that might be present in the clinical record that gives you a lot of detail about what the drug has actually done. We look at comparator compounds that might have relevance to it, either from a similar mechanism of action, or even something that can teach us a little bit about the disease state, even if it's a different mechanism of action, or even on a different sort of disease state that may also be relevant to it. And we do look at the experience of the leadership group for a company. And so you may have a small company, but perhaps their leadership are seasoned drug developers. And we feel that that would be something that would mitigate the relative short track record for a smaller company.
That's fair enough. And maybe if I can ask both you, Dan M. and Matthias, we've discussed some of the differences, but maybe what are some of the similarities? And also just what is the definition of success for a phase 2 study or a phase 3 study? Because I know with the different methodologies, there could be different definitions of what success is. Perhaps Dan M., I can pass over to you first.
Sure, thank you. And that's a really critical question for us. So we start by looking at what would be probably considered a more typical measure of success, and that's based on whatever the primary endpoint is of the trial. But we'll focus in a little bit more in detail and say, well, what does it really need in order to understand what's going to provide value? So what's going to be valuable for patients, and ultimately what's going to be valuable for the sponsors. So we'll look at that, we'll think a little bit about what comparators are showing, and we'll try to come up with an estimate of that. It might be relatively straightforward for a phase 3 study where you're looking at statistically significant success on the primary endpoint. On a phase 2 study, and we'll get this a little bit later when we talk about Vertex, it may be completely up in the air, and we have to determine ourselves what we consider to be a successful trial. But ultimately, it's what is the market going to say is successful, what are the sponsors going to say is successful, and what's going to be valuable for patients.
That's interesting because each of those components might see things a bit differently. Would you agree? Yeah.
I would agree. Although there's going to be a certain convergence ultimately, because if you don't have value to patients, then you're ultimately not going to be able to really differentiate from a product perspective.
Yeah, and that's fair. Matthias, what's the definition of success when you run your AI algorithm?
Yeah, for us, it's a trial reaching its primary endpoints. That's our definition of success. That's what the AI is really trained for, based on those 40,000 something trials. And you also asked about the similarities of the approaches. I mean, we're also using up to 200 features or so per trial, and then mentioned some features around the drugs, similarity to approved drugs, to other drugs, etc. And it's also really stuff that our AI looks at. that are features that we have in place, also pharmacology features, etc. So some of the data our AI is looking at is probably the same data or similar data as the Diviner colleagues are looking at. It's just a different perspective with different biases than human biases, and different challenges, of course, to the approaches. But I think there's also quite some similarity in it.
Mm-hmm. And then I guess the key difference is with AI, you can actually just really scan everything. So you can look at all the data, which humans really don't have the bandwidth to do.
Yeah, and our predictions are, of course, instantly available. I mean, you at the click of a button, you have the prediction available. It's, of course, a lot less work-intensive than an expert prediction.
Mm-hmm. And then maybe, Dan, can we get into a little bit more details about how you do the predictions then? Because the algorithm, well, it's not really an algorithm anymore, but there are algorithms involved, but it is quite different methodology, right?
Sure. So I think the core is really understanding what makes a good forecaster. And I really can't provide detail on that at this point. First, because we're not really there yet completely. We're sort of in a design, test, iterate type of mode. And so we're learning what makes a good forecaster, and we're identifying individuals who are good forecasters. But we're not there yet, and probably we're not going to be able to share it publicly when we are there. But what we do is we do a process that isn't a due diligence process because it's very focused and it's based on public information. We're trying to gather as much as we can about the endpoint that we care about. That's going to be technical success and value, as we mentioned. But what we are going to do is we're going to think about the full set of information from the critical trials. So when we talk about a trial, people will often focus on the point estimate of the primary outcome that is the top line result announced by a company. We might look at more information than that. We might try to say, what is the quality of the trial? Is there other information in the trial that makes it a little bit more shaky? Let me give you an example of that. So before we went through the 10 forecasts for this trial, we did a forecast on Idera Pharmaceuticals, and we looked at Bosotolimod, which was a metastatic melanoma compound. And what we found for that is, although their phase 2 trial, which was a single-arm trial, had produced what appeared to be a very, very positive signal that made a lot of people excited about the product, when we looked at that in detail, we saw some irregularities in how the data was put together and some of the parameters about how it was reported. And so we sort of tried to back that out. And when we backed it out, a lot of the difference from historical controls disappeared. And so we take the data that's in the trial and we try to qualify it and understand a lot more about it. And this is a very dicey proposition. You know, essentially we're looking at the data and we're saying, what can we read into it? If we came at it from a biased perspective, then you can obviously manipulate it how you choose. And that's why people always go to the pre-specified endpoint. But we're trying to approach it from a getting to truth perspective. What does the trial really show? And we feel that you need to look at all the information in the trial in order to get there. So I think that that's something that's a little bit different about our process. We're really trying to dig into the data and see what it really tells us about the true value of the compound.
Okay, well, thank you for that. I think maybe what we can do now is go into maybe more examples, because there are 10 trials that both teams predicted. There were some where there were similarities in predictions in terms of outcomes, and then there were some where there were differences. And maybe what we can take a look at is the one for Sage and Biogen's drug, Sage 217, for depression. But before we get into how the two different teams predicted that, maybe if I can pass it over to Dan C. to walk us through what the drug is and what the trial is about, just to give everyone a bit of a background on this.
Yeah, sure. This is a competition after all, so we have to do some judging. And thankfully, for the timing of this webinar today and this panel discussion, we actually do have one of the trials where we have a reporting of a primary endpoint. And this is Sage's antidepressants, Sage 217, in the phase 3 waterfall trial. Those of you that follow depression drug development, you may be familiar with the difficulty of running a pivotal trial. It's almost more often than not, antidepressants that reach the market will do so despite having failed pivotal trials. So a negative result doesn't necessarily mean lack of success. This is also a really interesting case study because this is a drug with a new mechanism, GABA-A, but there is a precedent from an intravenous formulation, brexanolone, which has reached the market for a slightly different indication. And we also have all of the phase 2 data for this drug. So it's a very, very data-rich example, and we have a huge divergence of opinions from investors, from the markets, and even with the clinical trial, which reached its primary endpoints, there was a significant improvement in HAM-D depression rating scale relative to placebo after 15 days, 1.7 point change. The market reacted negatively. The sentiment was that, okay, there was a significant reduction, but that the difference from placebo was waning over time. The expectations were higher. Meeting the primary endpoint was not enough. So really, really interesting example. We can speak to the results of that. So Divina, congratulations. This was one in which you got right with a pretty confident 76% prediction of success. And Molecular Health, well, you were on the fence there, but similarly, I wouldn't say this was a fail at all because, the market reacted negatively to this. The trial was not a slam dunk by any means. Yes, it's a successful trial and it met its primary endpoint. So I suppose, given the wealth of data that we have and knowing now that the trial result has been reported, I'd like to punt this over to the Molecular Health team. How did you feel that your AI handled this particular trial prediction? And do you think this is one in which you can learn from and change your approach, or do you think it validates your prediction?
Yeah, thanks. I think overall, with 39% probability of success, it's a prediction with a rather low confidence level, and that's also something we'd see in our software. So we have typically a true negative fraction of those of similar trials of 64%. So we'll be wrong in 35% of the cases. So it's more tendency towards failure we had. And I looked at the factors for potential success as well as for potential failure. What our AI considered as potential success factors actually is a rather high number of approved drugs in the indication, several fast track approved drugs in the indication, as well as the clinical operations side of the trial. For example, the number of sites, the number of countries. So it's just a single country in that case. Those were seen as positive factors. And there are some negative factors, of course. The drug itself has a rather low similarity to already approved drugs. That's one of the main negative factors the AI saw. And there are additional factors like a pretty high log P compared to other drugs here, and also still limited data from clinical trials for this drug compared to other drugs in the therapeutic area. And it's overall result in a prediction that tended towards failure. And you're totally right. It's technically it's a success. So from our definition of what we predict, we were unfortunately wrong. But I think it's really an interesting one for discussion since, yes, it met its primary endpoints, but yeah.
But it barely met its primary endpoint, right?
Yeah.
Yeah.
And I think that's one of those trials, I think, where it's really a good perspective, an additional perspective that you can look at. So to identify additional factors, to get an additional opinion. So in this case, a more negative one. But I think it's really could be incorporated into decision making, although we were here kind of wrong.
But you weren't that far off because some of your predictions are really low for some of the trials. And in this instance, it was about 40%, right? Or 39%. Yeah. So
that's a trial we would typically, when we present our application to others, we tend to say that's a rather inconclusive area of probability of success. But as we predict your success or failure, the AI says it's a potential failure.
Yeah. And Dan M., what is your take on this trial? And you predicted that it would be successful, but how successful did you think it would be?
So I'm really glad that we're talking about this one because I thought it was a very interesting example. And it's a very complex area. And as Daniel mentioned, depression is a very hard area to do studies in. There's a lot of population heterogeneity. There's a placebo effect. The HAM-D scale itself is highly variable. And so you can get many failed trials and still end up coming to market. We really looked at a couple of different things. We looked at the trials itself, and then we looked at what we called a success within this indication. So from a trial perspective, it was actually a wealth of information, enough such that we used that as the primary means of sort of trying to understand this molecule rather than trying to get at something like, do we believe in this mechanism of action? Is a GABA mechanism a reasonable approach to treating depression? There really were three main trials that we looked at. The phase 2 trial had a tremendous difference from placebo, but it was a small trial. So it showed a 7-point difference from placebo on the HAM-D scale, which is far in excess of what most of the antidepressant drugs on the market, which are more in the 2-point range, are able to do. So people were very excited about that. But I said before that we tried to look at the studies themselves. And so when you looked at that study, what you saw was actually that at the pre-specified endpoint of 15-day response, you saw the 7-point difference. But if you looked at earlier time points or later time points, it was a much more moderate benefit. So if you really tried to characterize the study, the 7-point difference looks a little bit more like a fluke than it looks like the true benefit provided by the drug. But there did seem to be a benefit provided by the drug. It seemed more in the maybe 4 to 5-point range. So how do you characterize it? That's a judgment question. The second main trial was a failed pivotal trial, the Mountain trial, and that was a much larger trial, intended again to be a pivotal trial. At its baseline, that trial actually had a difference from placebo of about 1.4 points. So not statistically significant, but if you look a little bit earlier, it was statistically significant. So at earlier time points, you saw a couple of points different from placebo. The clinical trial population for that study was somewhat different from the phase 2 study in that the baseline utilization of antidepressants was much higher than in the phase 2 study. So you could think about those as a little bit different populations, and you could say, well, what do we learn from combining the information from those two trials? The third trial we looked at was actually the Robin trial, which is in a somewhat different indication. It was in postpartum depression. And in that trial, which was intermediate between the two as far as the size of the trial, it was about a 4-point difference or 4.2-point difference on the HAM-D scale. So how do you pull this all together? We had 3 trials. All of them seemed to show some sort of a benefit for the drug arm. So we believed that the drug was active, but how do you try to combine these factors? And there were other factors that I don't have time to really dive into about the individual trials that might make them more believable, less believable, increase the expected benefit or not. It wouldn't be appropriate in a classic sense to do a meta-analysis on these 3 trials. We had a small number of trials. You've got differences in base population. You have differences in other factors about the trials. But you do want to try to pull together the information from the trials and try to get to a global understanding of what is truth about this compound. And so we did some of that. And we said, we think it's effective, but what is the effect size? And the confidence interval on that is pretty broad. It's something like 1.25 points on the HAM-D scale to about 4 points. And what do we think is going to be value is the second question. So there are a lot of benefits that this kind of a drug could bring. So you're looking at a product that has a faster onset of action than most classic antidepressants, but not as fast as ketamine. You're looking at a product that has a persistent benefit after patients are treated. It might be good for a little bit more episodic treatment than some of the classic antidepressants. And you're looking at a different mechanism of action. So maybe it's going to work in a population that is not as much benefited by some of the other antidepressants. So these are all things you could consider, but what you have is a trial that's looking at a more classic antidepressant type of endpoint. And so if you come on the market, you're competing against a lot of generic products that are providing about 2 points of benefit on a HAM-D scale. And so we looked at that and said, to really be able to compete with those, you probably want to have a value that's more in the 3-point difference on the HAM-D scale. If you have 2 points, we figured that was about what you would need to have statistically significant separation. That would be a successful trial, but it wouldn't be perceived all that well by the marketplace overall. And so you'd have to then do additional studies to prove that you could be beneficial in a subpopulation. So we forecasted at the 2-point level, we forecasted at the 3-point level, and then we threw in the possibility that there might be adverse events that come up that might limit utilization. That was particularly relevant because they increased the dosage in this trial from 20 or 30 milligrams in some of the other studies to about 50 milligrams. So we pulled it all together, and then it was given to the individual forecasters, we call them diviners, and that was how we came up with the 76% range. But we divided it up into different scenarios, and some of them were less positive, and some of them were more positive. So, 2-point difference, which we considered more likely than a 3-point difference, was something that would not be, we thought, all that exciting to the marketplace.
Okay. Thank you for that detailed explanation. That is interesting. It is such an interesting market, and it's interesting that the trial was positive and the market reacted to it negatively. So maybe Dan C, perhaps, if you want to comment on any sort of last sort of notes about that trial, or what else we should be looking out for?
It's an interesting case study, and I think we can all learn from each new readout in depression and how different mechanisms work, and certainly from the detailed explanation of how the diviners consider each scenario. There is one other one other clinical trial that I wanted to get onto, because it's polar opposite from the depression markets, where we have hundreds of approved drugs, seemingly, so many clinical trials, but focus on a much rarer disease and a much smaller clinical trial. And that's one, this is also a clinical trial where both Molecular Health's AI algorithm and the diviners agreed. And that's Vertex's clinical trial in a disease called focal segmental glomerulosclerosis, FSGS. This is a disease where we don't have any approved drugs. There's no pipeline whatsoever. There is some use of drugs like hypertension drugs to manage symptoms. But for the first time, we're trying to develop specific drugs for these specific patients. Kidney disorder, commonly with scarring, and some of those symptoms include proteinuria. And the endpoint that these FSGS drugs are trying to improve at the moment is proteinuria levels. So if we go back to the contest and the prediction, if I just refer to that. So on the diviner side, we have a 74% likelihood of success. And in the Molecular Health, we have a 67%. Very, very tightly bound, which is, and so clearly there's a lot of encouragement and hope for these FSGS patients, that there is hopefully a specific treatment being brought with a high degree of success to the market. We focused a lot on the differences between AI algorithms and humans, but perhaps let's focus on some of the similarities and what leads you both to consider the strong potential that this drug will progress through this clinical trial. Matthias, Friedrich, what are your perspectives here?
Yeah, I mean, again, it's with the 67% likely probability of success. It's still in a it tends towards success. It's not a very strong prediction towards success. And our key factors were the biomarker, which really was the key factor. So a known biomarker that is always a biomarker part of studies, always an indicator of some mechanisms are understood and there is a clear target. And other factors were the drug itself, the trial design, and other factors of the sponsor that were considered as positive. But those mostly had lower impact. So it's really mainly the biomarker that drove this towards success. There's a few features with with potential negative impact. So the number of sites is considered as potentially negative here by our AI. With 41 sites, seems to be a pretty high number of sites for a phase 2 trial. Also, the duration was seen kind of skeptical by the AI. but not really with very strong impact. And overall, yeah, this resulted in a prediction that tends towards success, but as said, not with a very high confidence level. So the true positive fraction in such a probability of success area is 67% of similar trials where we had no outcome available. Yeah.
I should also say that this being a rare disease and being a very early phase 2 trial, I believe this is an open label trial. So in the strictest sense, there isn't that measure of success that your algorithm would typically rely on, which is meeting a primary endpoint. So in this sense, how would you, what is that success criteria for your algorithm here?
I mean, the algorithm is trained on 40,000 something trials where we have known outcomes for. So many of them actually we have curated outcomes, since negative outcomes tend to be underreported. And that's what we train them on. So the algorithm itself, it does not need or does not look at the individual primary endpoints of the study, but it basically predicts that this will be considered as a success. That's basically what it predicts.
And I really like the comment about biomarkers, primarily because it agrees with some of the published research that we've put out there at Informa. But yeah, just to say, I suppose each biomarker has varying utility, and it's very much in the spotlight with the utility of amyloid as a biomarker. And it'd be interesting to know whether in the Alzheimer's disease setting, the positive emphasis that amyloid would have on your predictions. But more generally, our kind of observation across all biomarkers and whether they're used to enrich clinical trial design is that it typically raises the success of an individual drug program 2 to 3 times over the baseline. So, we would just always reinforce that message. Even if you're targeting, if you're targeting a particular disease, really refine your patient populations. And in this case, we have, the mechanism of the drug is very much linked to the biomarker in investigation, which is apolipoprotein L1. So the Diviner approach to a trial where there isn't a whole lot of precedent. So how do your experts kind of find that information to form a balanced judged opinion? Dan, M?
So very good question. In this case, there's a lot more uncertainty, I think, around our prediction than there was about ones where there's more data. To the question that was asked a moment ago about what do we define as success in that trial, that was a challenge for us just starting out. We looked at this as a disease in which there's a high unmet need, and we essentially looked at the primary marker, the primary endpoint for this study, which is UPCR, and we said that since there's an expectation that there's continuing decline on that measure, that anything that at least stabilized it would be considered success for this early stage trial where there's not a lot of unmet need. So from a perspective of what we looked at, we actually were fairly similar to what Molecular Health was saying with regard to this particular trial, because we put a lot of emphasis on the fact that there was a solid genetic linkage for this particular disease, and that was important both because it validated the target and because it allowed a more homogeneous trial population, which would, we felt, increase the probability of success quite significantly. There's preclinical data for this. It's hard to understand how predictive it was in the absence of a lot of depth on that really hasn't occurred yet within this disease state. The one thing that I would throw out that is sort of interesting to us is that Vertex's CEO is actually a nephrologist, a physician trained in that area. And so we looked at that and we thought about whether that was a positive or a negative sign. On the positive sign, it would give more scrutiny to this sort of an area. On the negative side, it might create more impetus to bring something in that area forward because it might be at some particular interest there. We did say that it would be important from a managing of the clinical trial perspective in that there were certain unknowns as far as how they selected their patient population, how they evaluated patients, and we felt that they had the expertise in order to try to bring this trial successfully forward. That's kind of how we ended up at, assessing this as more of a high probability than a low probability. But again, this is something there's a lot of uncertainty around.
Okay. Thank you for that. And I guess we'll be on the lookout for that trial data to read out before the next event at BioFutures in October. I think we've actually run out of time. So I'm going to wrap things up by asking you all just, we've seen similarities and differences between the two approaches. Clearly, there isn't a right answer anywhere until the trial data reads out, but even then, it's murky sometimes. So my question to you, and maybe I'll go to you first, Dancy, as sort of the more independent panelist here, as to, now that we've heard both sides, both methodologies, where they converge and where they diverge, do we have to pick one or the other? And what would your recommendations be?
Well, you set me up right there. I'd say triangulate, use all of the information you can that's available to you, understand the methodology, understand the sourcing, and judge for yourself, use your own critical. So whether that's applying human intelligence to human intelligence or human intelligence to AI, naturally, it sounds like what I'm suggesting is almost that wisdom of the crowds approach anyway, where you take an average of an average and come to your own opinion. So absolutely, there is so much we can learn from AI, and the more we develop it, the better it's going to get. So I would be hesitant to ever suggest that, to disregard any of the really strictly negative predictions that they're making. I think there's a lot of value in understanding why those predictions are so low. And then similarly, you'd want to know why the collective intelligence of experts is also, I think there's a lot of value in understanding that too. So yeah, I mean, the obvious answer is a bit of both. And hopefully over time, the AI gets better, and hopefully over time, our experts become even more expert. And then we find the true answer.
Thanks, Dan. And maybe if I can pass it over to Friedrich or Matthias for your take on this.
I think we with AI, I mean, as you said, I mean, it will develop further and further and improve. And really the key to good predictions is data, unbiased data, clean data, complete data, and also sufficient quantity to train your algorithms on. And that's really the challenge. I mean, some data that would be really beneficial for training is often not available in the right quality or quantity. So that's definitely something that can be improved on. And often still needs manual curation, even upfront AI predictions. So still also humans are highly involved in that side. And one thing is, I mean, also in other applications of AI, I mean, you have driving assistants in your cars and you don't blindly rely on it, but you're still kind of an expert of driving your car. And so you're an expert also, if you're an expert in the clinical trial area, then, I mean, you will see those results. We are very transparent with what are the success and failure drivers. And so you can really make your own informed decision whether you want to incorporate it into your decision making or not. And you always have an additional perspective that's instantly available. Mm-hmm. And also, since we have those available for all the trials, pretty much all, like 68,000 trials or so, you can also, this allows also for really other applications. So you can look at complete portfolios of trials, etc. So something that with the human expert-based method, you're very likely not to really be able to establish.
Thank you.
Let me make one additional, completely different comment. I think one of the biggest challenges in healthcare is the white spots or the unknowns that we have. I mean, I think the big difference to a GPS system, for example, is, I mean, the globe is known to be round, and we both basically can identify every spot. In healthcare, the problem is we don't know what we don't know sometimes. And I think what AI can do, at least in certain cases, and this is why 100% accuracy cannot be expected in the near future, is I think AI can, in ways we, with our brain, cannot always follow, try to close some of these white spots by triangulating, and triangulating is only simplification for n factoring, so to say, what we don't know. I mean, even since the system is trained on what we do know, even that system is error prone. So it's a different bias, of course, than humans have. And the art will be, and someone said that before, the art will be probably to have systems like this in the hand of trained users who, coming from both ends, bringing the best of both worlds together. But let's not do the conclusion here at this time today. But basically, this is my firm belief, at least in the year 2021.
Yeah, and I wholeheartedly agree with you. But I'm going to pass it over to the Diviner team for their take on this as well.
Thanks, Lee. Yeah, this is really, really interesting, and it will continue to be as we see how these trials read out and as we do more. But it is absolutely, we believe, going to be a combination of machines and humans that produce the best result. As we said, we're just in the early stages with Diviner. We will scale our network with more forecasters. We will supply them with more analytics to make better decisions on. It'll be that combination that's important. If this intrigues you and you're interested in providing industry expertise or forecasting skills, go to diviner.co. We are looking for people that can expand our forecasting network. It isn't going to be easy to predict trial outcomes and stock prices, but that's the mission. And it really will be a combination of humans and AI.
Thanks, Mark. Thank you all for your time today for a really thought-provoking discussion. I hope our members and the viewers enjoyed this as well. And with that, I'm going to pass it over to Sarah for any questions that we might have.
Yeah. Hey, thank you. Thank you very much, Lee. And yes, a few have come in. So very first one, and this one I think goes over to Friedrich and Matthias. I've seen a questioner writes, I've seen a lot of reports of highly flawed data sets that are foundational to machine learning and artificial intelligence applications. So how does DataOM succeed where others have not?
So what we are doing is we also have a lot of human experts involved in feature engineering, cleaning of the data, making all those data and different ontologies compatible, mapping of drugs and all that. So that's things that also the humans do, but at a much later phase. So also Diviner does probably, but at a much later phase. And we do this for getting the most reliable data possible for our algorithms. So we also involve manual curation where needed, for example, for biomarkers, for clinical trial outcomes, etc. And so that's really what we do to minimize or reduce those flaws. But I mean, I think we discussed this earlier. I mean, there's always biases in data. There's always clean and incomplete data. We do imputations of data if there's data missing. We have different predictive models, some of which, for example, can cope with certain types of data missing better than others. And so that's basically how we challenge those or try to reduce those issues.
Okay, Friedrich, did you want to add anything to that, or shall we move on to the next question?
No, thank you. It's good.
Okay, so the next question comes in from another participant who's from a pharma company, and they write, for the potential success criteria, are you considering the therapeutic area, CNS, CV, metabolism, immuno-oncology, rare diseases? Some areas such as CNS, neurodegenerative diseases have the lowest probability of success. Some endpoints or scoring are sometimes are highly variable and subjective. Do you integrate a factor per therapeutic area? And I think that's for both of you. So it would be Matthias and Dan.
So our simple answer is yes, we do.
Okay, that was a very big question and a very brief answer. Thank you very much for that.
We consider, of course, data from therapeutic areas, as there are therapeutic areas with much higher likelihood of success than others. And yeah, we have those data incorporated.
Dan M?
So we look at each event that we're trying to forecast independently and try to come up with our understanding of what the success is likely to be given the endpoints for the particular trial. And what you'll often see when you look at therapeutic areas is you might end up mixing things that have higher and lower probabilities of success. So you have to take that a little bit cautiously. But we do think that there's going to be higher tendencies towards success in some areas and lower in other areas. We're just getting started. And so we haven't been emphasizing some of what might be considered anchors from a bias perspective as we start out doing our forecasting process. But there is a rationale for pulling those in because it allows people to focus a little bit more and gives you more confidence around where you're calibrating your overall probability of success, which is something we'll have to look at as we get more forecasts. And this is really a great opportunity for us to do things collaboratively with an AI type of approach, because it can give you a really good idea of what expectation is. And then you move off of that and say, how does this differ from norms? Is it better? Is it worse?
Sarah, if I can jump in on that question as well. Between therapy areas, there can be like a 7-fold difference in overall likelihood of reaching the market. I think haemophilia and hematology is really, really successful. But it's important, this isn't the only thing we can consider. If all of our investment decisions are driven strongly and our clinical trial design decisions are driven wholly by progressing drugs onto the markets, we're just going to be reinforcing traditional biases and are not really addressing unmet need. There will be therapeutic challenges where you need to be taking those, well, whether you call it a moonshot or long shots, and absolutely there is critical importance for predicting clinical trial success and designing clinical trials in a way that's going to maximize the likelihood of showing a clinical trial benefits. But I think it has to be one of many aspects that's considered, because otherwise, these rare diseases, these patients out there with no treatment alternatives, they will be increasingly be forgotten. So importance, but not the be all and end all.
Right.
What you can do also, just one more comment, sorry, what you can also do with our software is actually you can look at self-defined sets of trials. So, for example, you can define a set of trials from a therapeutic area, and then you can also see with respect to probability of success and other factors, where does your trial of interest stand in the respective therapeutic area. That's also a perspective we can provide, and that may help to judge on a trial.
Yeah, one of our, and I think this question feeds into everything that you've just said. The questioner from a pharma company wants to know what are the most difficult therapeutic areas to predict. And I think that's a question perhaps for AI right now and also for the human side. So Matthias, Frederick?
Actually, I could pull this out of the database, but I don't have that information readily available. I'm happy to deliver it post the meeting.
Okay, which is the most difficult?
Yeah.
Okay. Relying on the push of a button, huh?
Let me make one additional comment. I mean, we have asked the question, for example, whether novel therapies like gene therapy or whatever would have a worse outcome or predictivity. So we found not. So what I want to say is, I don't know the question to that answer, but as I said before, I mean, the way AI works is probably does not have a bias or favorite in therapeutic areas. I mean, we can look at outcomes, so to say, which therapeutic areas are more or less successful. As far as I remember, we don't see big differences. So the technology seems to, and I can't explain why, but seems to nibble that out.
In terms of accuracy, we have pretty even accuracy across all therapeutic areas. It may be that in one therapeutic area we have more negative predictions, while in others we have more positive predictions, and that's what I can pull out, and then we have the kind of the facts available. But overall, we have pretty even accuracies.
So go ahead.
From our
I wanted to switch over to who's speaking.
This is Dan.
Oh, okay. I was going to say, let's move to the diviner team. So Dan, you're right on the spot here.
From our perspective, I think that it also is difficult for us to understand at this point where we're going to have the best predictive success, but we actually seek out areas where there's a tremendous amount of uncertainty. Because I think if you're looking at relatively precedented mechanisms and drugs that don't differentiate a huge amount, there's a lot easier ability to come up with accurate forecasts, but we also think that that's less an area of value for us and potentially less an area of value for patients. So we look at the really innovative stuff, and we think that that might be where we differentiate more.
And that may be
Okay.
where it diverge a bit too, just because as we see more gene therapies and more novel treatment modalities move into the rarer diseases, there is less data in those diseases. So this is where I think the combination, just because there isn't much data, sometimes the endpoints are brand new, the trials are brand new, everything's brand new in these for these rare diseases. And even, you have people questioning whether the endpoints make sense or not. This is where I think the human side comes into play and we can help each other out as well.
Right. Thank you. Thank you for that. Moving on to some other questions, because I know our time is going to be up shortly. One of our questioners asks, assuming the assessment of various factors make weighted contributions to the probability of success score, how is that weighting determined? Does that weighting vary with disease, class, or type of therapeutic, or other considerations? I think this is a question equally for the human side diviner as well as for the AI side, molecular health. Dan M?
Sure. We don't do a weighting of factors per se. We try to allow where there's good expert information to allow that to win out, and where there isn't, we allow judgment, which is really about the integration question. So, as I mentioned before, with SAGE, you can do a little bit of a crude meta-analysis on it, but if you're trying to pull together other sorts of things, it's much more judgment-based. So we don't have a weighting per se.
Okay, thank you.
What's interesting about bringing a diverse group of people together with different backgrounds, some in the therapy area, some in other therapy areas, is they will spot the things that should be really important. Even if it's in a rare area where there's not a lot of data, it's that diversity of perspective. They will raise the issues that are most important, and then their peers all within the group can assess. So it's really a helpful way to apply human judgment.
Okay, thank you for that. Friedrich and Matthias, for the AI side.
So what I mentioned earlier, we have not just one single predictive model, but we have overall 16 predictive models. One thing is very obvious. I mean, depending on trial phase, prediction of trials is completely different. It's phase 1 trial is not really comparable to phase 3 trial, for example, and we predict across all trial phases. So we have different models optimized for the phases. We also have models optimized for other aspects that I cannot really go into further detail. But those have used different sets of features. And if, for example, trials don't have some features available, I mean, we might, we can still do predictions. They might, of course, be less reliable, but it's still predictable. But with our broad coverage of models, we really try to have an optimized model for all the various areas available.
I mean, let me add one comment, and that goes into the systematics of the gradient boosting systems. I mean, AI. Let's not step here into details of AI, but adding one comment. I mean, depending on the question and depending on the data and depending on the challenge that the question to the AI system has, it calibrates and gives weights by itself. I mean, I could explain now, for example, leave one feature out method, for example, how that works. So long story short, we don't give weights into the system. The system on the question gives the weights by itself. But we can look later on, if we go into the feature analysis, what the system did and try to understand why.
Okay, thank you. We have about 4 minutes left, and so let's see if we can get through this next question, fairly quickly. And then our last one is a big question. With increased enforcement of clinical trial results publishing, what are the implications for machine learning and algorithm approach to forecasting? And maybe Lee and Chancellor, this is something that you can also comment on.
Well, very quick opinion is it's good. The more we know, the more that's public domain, the more data is available, then the more that data can be used to reinforce good decision making. bad data or invisible data doesn't help anyone. So it will help move the field forward. And the same with any type of data, whether that's health records or, but yeah, certainly clinical trial reporting is important to capture.
As Matthias said earlier, a lot of negative trials don't get published. And so the data is actually quite skewed. So if everything does get published, it actually does help learn more what contributes to negative factors.
Yeah.
Right, right. But if you don't know what the negative results are.
Yeah. I mean, oftentimes, you may even have to like define rules, like if a trial is completed and there's nothing published for whatever 2 years, then it's probably a failure. I mean, you really have to go to that level if there's no really results published, negative results published. And it will be really very helpful and very important for improvements in the machine learning area. And for the whole industry,
there's not really open source, so to say, for clinical trial results. So let's move on to our next question. And this one goes back to molecular health. Sorry, Diviner. The question is, to what extent is your model taking in data about clinical trial sites and primary investigator for the trial in predicting whether the trial will fail or succeed?
So we have, I think in those areas, roughly 10 of our 200 features or so. I mean, they're taken into account. So not the individual sites, but more on the number of sites. So like if it looks like a typical trial or if it looks kind of special from that perspective. And of course, they can have a significant impact. But it's always really the combination of all the features that really then drives the prediction towards failure or success. Even if one of those factors is considered as very negative for a trial, it doesn't mean it has a super low probability of success automatically.
So can I make one comment as well? And this is the Vertex trial, because Matthias brought up that there was, I think, 41 sites for that trial, and that was perhaps a negative feature for a trial that size. We actually saw that as well. And given that there are only 10 patients in the trial, and that means they're recruiting a quarter of a person at each of the sites, we actually think of that as a little bit more of a positive feature, because when you look at that and you say, well, what are they doing? They're planning for the future. And so they actually have confidence in the product, and that's perhaps more positive than it is negative.
Interesting thought, yeah.
Okay.
Yeah, but then can I make one comment?
Yeah.
It's interesting because, I mean, you're right from the human brain thinking. The AI technology has only one task, make a decision on that particular trial. I like your comment, but AI, I think, doesn't mind.
And we actually see something quite different just because we have the SiteLine suite of products where we do look at trial sites and investigators and their experience in recruiting patients. So the more sites they open up could be because they can't find the patients. So they need to open up more trial sites to try to find those patients. And this is a rare disease, so it's actually quite typical for that.
It's an example where the more data that's available, the better your decision is going to be, right? And if data's not there, you don't know, and you're guessing. Yeah.
Yeah.
Okay, so a follow-on to that question, and this will be the last one before I ask all of you to wrap up. If the best and machine working together, I think this is a prediction for the future, MI, molecular health and diviner, how interpretable, explainable are the predictions your AI is making? Can human interrogate the rationale behind the prediction, or is that a black box model? So this is for Matthias and for Friedrich.
It's clearly not a black box model. We have a whole feature exploration module that provides you access to the features and shows you the impact of the features on a prediction, also some what ifs, for example, like how would a probability of success change if the sites were 60 instead of 40 or something, or if the trial had a biomarker versus no biomarker and all that. And we really provide all this information, also the most impactful feature groups. And so it's not a black box. And I think that really how we developed, and the software and its user interface is really made for interaction of humans with AI.
Yeah. In addition, I mean, the next step would be a trial composer in addition to a trial analyzer, because of what Matthias just said.
Okay, wonderful. That's our last question. And with that, I'd like to go around to everybody and have you wrap up. And I think in this case, we'll go to Friedrich, then we'll go to Mark and Lee and Dan. Unless Matthias and Dan also want to comment, we are really quite out of time. So Friedrich?
Fine for me.
Yeah, I mean, very quickly, I mean, I think everything was more or less said. I mean, the future will be, and I'm bet on this, the future will be a combination of both, and such technologies will play a role because the complexity of human biology, disease understanding, chemical space is so diverse that only machine can support man in this. But a machine without the question and the use case don't make much sense. So that will be the future. And I think you're kicking off, Sarah, you and your team, you're kicking off that awareness process. And I'm really looking forward to the next meetings here in that group. I mean, it's a real kickoff situation.
In person in New York City. So we look forward to seeing everybody there. Mark, brief comments?
Sure. Thanks, Sarah, for putting this together and everybody. And we're excited to do the second round in New York and see how it goes. And we'd love to hear more ideas from the attendees about how you can get involved in trying your hand and seeing if you can compete with the two groups.
So wonderful. Thank you for that. Lee?
I don't have much to say except that I think this was great in that we're bringing something to the forefront, which is how do we, maximize what we can do with AI and with humans, and how perhaps we can best combine the two together to make things, a lot more accurate in the future. So this was great. Thank you. And I look forward to the next round.
New York, everybody's coming to New York, so we're looking forward to having you all there. And I do want to thank you all for the discussion. I mean, part of what we are committed to doing is to help advance healthcare and delivering that, to people. This is part of that process. So I think you're very brave to take this on, and let's see what happens in October. Before we sign off, everybody, I do want to thank Terea and our previous virtual salon series sponsors and our founding sponsors for BioFuture for their ongoing support. I'd like to thank the audience, and please, as you leave, there will be a survey that you'll be invited to participate in. And I'd like to ask everybody to join us for our next virtual salon, which is fundraising strategies for European biopharma, which will take place on July 8th. Until then, everybody, stay safe, avoid all those variants, make sure you get your shots if you haven't gotten them. Signing off.
Thank you.
Bye now.
Thank you.